So I'd like to talk to us today about next-generation psychedelics and entactogens and look at both the compounds that Dr. Jetley just introduced us to and think about how can those actually be improved for use in the clinical setting, and show you some examples of how at Adolescent we've actually attempted to do exactly that, and I'll show you the progress that we're making on this goal. So just to give you a short background about myself so that you know who's talking to you today, I have a different background than I think a lot of people in attendance here. I'm not a clinician, but I'm a researcher. So I'm the Chief Scientific Officer and co-founder of Mycin Innovations Group. We are dedicated to improving psychedelics for use in the clinical setting, not in a recreational setting at all, really tailoring these for use by clinicians for better efficacy with patients. We're interested in both psychedelic and entactogenic compounds, and we really focus on taking these drugs from concept all the way through into clinical trials and further into regulatory approval, although we haven't gotten that far down the pipeline just yet. So my background is really in developmental biology. I originally was an academic before moving into the commercial space. In that work, I really developed a skill set in drug design, genomics, and genetic engineering. And since leaving academia, I've been applying that to a commercial setting and really focusing on intellectual property covering these exact concepts. So I have about 5 patents that have been granted at the moment, and then a continual sort of docket of 12 plus applications in process. So this is really how I've been focusing my time since leaving academia. Excellent. So why psychedelics and entactogens? Why do we care about these for use in a clinical setting? There's really a few points that I think can be either the most important, depending on the context and the indication that we're talking about, or can be a combination of these different points, depending on which indications we're trying to treat and exactly the different patients that we're dealing with. So firstly, these drugs are very strongly demonstrated to increase neuroplasticity. This is a really interesting property of these drugs and is not common in many other areas of pharmacology. And so the idea of being able to leverage that in a mental health setting is something that I think is very intriguing. Secondly, like Dr. Jetley touched on, some of these really lower the defense in therapy. So patients that have a high level of being walled off against their therapist have a very hard time connecting and actually thinking through what's causing their current condition. This really lowers that inhibition, and you see, time and time again from some of these clinical trials reported that, patients could immediately connect with a therapist under the influence of these compounds, where in the past, for years of therapy, they really struggled with that, and that was really an impediment to them actually moving forward and seeing success in therapy. Thirdly, there's an interesting property of these drugs where you can have what's usually called an epiphany type experience or a mystical type experience. A lot of the work at Johns Hopkins has really shown, especially in the smoking cessation work that Dr. Matt Johnson did, that there's very strong correlation between patients having a, quote, mystical experience or this very strong epiphany type experience and actually being successful in quitting smoking. And so it's an interesting thing to think about in therapy, where you can set up a course of therapy where you're, priming the patient for having this sort of experience, and then use that as a break point, if you will, to then follow up from. And it allows the patient to really see there as a beginning and then an after as far as the course of therapy. It's just an interesting concept to think about that way. I think, like Dr. Jetley was saying as well, it's not absolutely necessary for every patient, but you can think about how it can be leveraged to really improve outcomes. And then lastly, the durability of the treatments that we're seeing here are very, very intriguing. So we're talking about, 1, 2, or 3 drug sessions over the course of maybe a month or 2, and then seeing, again, in that smoking cessation work that I think is really a great example of how this could be effective, they have durability of over 2 years with patients that were prior pack-a-day, 30-year dedicated smokers and had tried multiple times to quit in the past, and they had an 80% success rate for quitting for up to a 2-year period, which is kind of the length of what they've been able to monitor so far. So it does really suggest that this is highly durable. That is a big sort of comparators to something like ketamine that's already being used, especially in North America, fairly widely for mental health purposes, the durability doesn't really seem to be there. So these, I think, can all really be leveraged to really improve outcomes for patients. So potential indications that have really kind of risen to the top for the interest of researchers. So with MDMA, this is like we've been talking about, this is the street drug ecstasy, but MDMA has shown incredibly strong results for PTSD in the MAPS trials. We've seen 80+% success rate for treatment refractory PTSD. There's also really strong interest in anxiety. I'll go into a little bit of how this drug actually works in a minute, but the potential to have an alternate to benzodiazepines, an alternate to other anxiety treatments is really very interesting. And then thirdly, before this drug was actually scheduled, it was very commonly used for relationship therapy, for couples therapy, this sort of thing. And you can imagine how a drug that increases empathy, lowers sort of a barrier to being walled off to the people that you're communicating with, you can imagine how that could be really leveraged for marriage counseling, relationship counseling, etc. So the next substance that's really seen a lot of interest is psilocybin and psilocin. So psilocybin is the prodrug of psilocin, so we're really talking about the active psilocin here. There's been really strong results out of Imperial College London. Compass Pathways is now trying to commercialize on this, but they've seen really durable reduction in depression symptoms in those patients. Addiction, like I was just mentioning, Hopkins has done really excellent work on smoking cessation. They've partnered with other universities to look at alcohol use disorder. There's strong interest in opioid use disorder, methamphetamine use, et cetera. It really seems that the mechanism that you're working with there is not specific to the substance of abuse. I know that some of the individuals at Imperial College London are also looking at behavioral addictions, such as gambling. So it really seems like it gives the therapist the ability to help the patient sort of re-architect how they think of their substance or behavior that's causing them the addiction, and they lose their excitedness, their desire to use those substances after that style of therapy. Finally, with psilocybin, there's a lot of interest in and success with end-of-life anxiety, people that are terminally ill, especially younger cancer patients that are having a hard time processing the fact that they have a terminal illness and will soon pass away. This seems to really have a strong impact on them being able to accept that diagnosis and move forward without a huge level of daily anxiety. And so other indications of interest, there's been, because of the sort of both academic, clinical, and then popular nature of interest in these substances globally, there's been a lot of thinking about how do you actually then extrapolate this to other things. And because of the neuroplasticity, there's very strong interest for traumatic brain injury, other sorts of injuries such as stroke to neurological tissue. Again, because of the neuroplasticity, there's been interest from the Alzheimer's and dementia treatment side of things. I think that's something where certainly there's more concern to be had there as to how that would actually play out with the patient, but the ability to leverage that neuroplasticity, especially at a lower dosage than would be majorly psychologically active, could be very intriguing. And the neuroplasticity that we see with these substances does accumulate over time and over use. So you don't necessarily need to have a very large dose just to get that single property. And like I said, there's many more conditions that people are interested in looking at. How could this actually be useful because of the unique properties of these compounds? So what can be improved here? We have compounds that are either directly from nature or have been compounds of abuse that have never really been designed for use in a clinical setting. The use with patients and the use by clinicians was never at the forefront of why we have these compounds. And so there's some things that really stand out that are really either practical or looking at safety aspects of the compounds. So the time course here is a very big one. We have longer durations for these drugs. They're highly psychoactive at the relevant dosages, and so you need to have a lot of monitoring associated with that, and that becomes a burden both for health equity but also for cost to apply these out into the wider space. And so this is something that we can use pharmacology to actually address. We can, look at how would we lengthen the time course, we can look at how would we shorten the time course, and I'll show you some examples of how we've been focusing on shortening the time course for some of these compounds. So we also have receptor specificity. Usually when you're designing a drug, you want to know very nicely the mechanism that you're interacting with and then target that mechanism very precisely. And the thing with these compounds is they tend to be fairly broadly acting across the serotonin receptor family, and so you may have properties that are very good, but you also have a lot of side effects because of that. And so that's something that we can address again with pharmacology, and we can improve control and improve our knowledge of exactly what we're interacting with, and reduce some of this potential confusion based on working on multiple mechanisms at the same time. And then thirdly, there's a lot of variability in the response to the same dosage patient to patient, and even patient within the same patient time to first use to second use to third use. And so this is something that is pretty hard to deal with in the clinic. If you have a patient take a drug and then you're going to be trying to run some sort of therapy session overlapping with the effect of that drug, and the onset could be 15 minutes or a half hour, or it could be an hour and a half, it becomes pretty hard to schedule that. Also, it's difficult for the patients because they don't necessarily know what to expect. That can induce anxiety if the drug takes effect much more quickly than they're expecting, or takes a lot longer. It can lead to anticipation and anxiety type problems. This is also something that we can address. So the design strategy of how we've actually made these modifications is to firstly look at being very conservative with our modifications. So we've looked at basically how do we retain as much of the original properties as possible, while kind of in a point-wise way looking at improving those bullet points that I was just mentioning on the last slide. And so we've been very conservative in that, not trying to reinvent the kitchen sink, but make one change here or one change there, and then looking at that effect to try to get to those improvements that I was just describing. And so the process that we've done here is we first looked at modeling the active pharmacophore. So we've taken these original compounds, put them into computer modeling software, as well as the receptor that we're interested in looking at, and then we've modeled how the two are docking to each other. This gives us a really good idea of which part of the molecule is actually responsible for the effect that we're looking at there, and then also highlights the opposite side of the molecule, if you will, that we now have more freedom to operate and can make modifications and look at, how do we actually gain some of these properties while retaining what we started with. So usually what we do at that point is that gives us a location where we can make point-wise substitutions in the molecule. And so we'll look at, I'll show you exact examples here in a minute, but we'll look at the original structure and then say, can we replace a carbon atom here with, say, a nitrogen? Can we replace a oxygen with a sulfur? And what will that do to gaining some of these properties? And then the final way that we kind of move this to, actually having candidates that then we could move into further trials is we'll synthesize the compounds that we've made. So, that middle step, we end up with a family of compounds by kind of iteratively doing that strategy with the molecule. So we may have, 3, we may have 5, we may have 12 candidates that are all going after that same idea. We'll then synthesize all of them and then do in vitro testing to look at, well, does this change the receptor that it's binding to? Does this change its stability in a liver cell culture? These sorts of things. So we get a fairly good idea if we're hitting the goals that we actually started out for. So psilocybin and psilocin analogs. Psilocybin, the normal compound that you think of with this sort of category, is the prodrug for psilocin. Psilocin itself is very unstable, and so you can't easily make a prescription psilocin drug because it'll oxidize into a blue compound that's completely non-psychoactive. In nature, psilocybin is actually a kind of perfect prodrug in that the molecule, when it has this phosphate group, is very, very stable, and then ubiquitous enzymes, phosphatases, will cleave that enzyme off, or that phosphate off, and then render psilocin. However, if you're trying to make a very time-controlled, repeatable drug for clinical use, the fact that you have a prodrug and then need to have moderated by an enzymatic step to then end up with the actual active component, this is adding in an additional layer of variability patient to patient, and even that, like I was saying, the same patient just time to time to time, and then can be highly impacted by things like what they've eaten that day, their hydration level, functionality of their GI and liver enzymes themselves. So it just adds in a whole extra layer of variability. So that's something that can be addressed. The main activity of psilocybin is, like I was saying, it hits almost all of the serotonin receptors, but the primary activity is via 5-HT2A, the sort of classical psychedelic receptor, and 5-HT1A. Like Rakesh was mentioning, that's also relevant at 5-HT2C. You can kind of make a descending list of the relevant receptors that it's hitting. Issues, though, with that is that it hits the 5-HT2B receptor, and this is something I'll go into in a little bit more detail, but this is highly undesirable property for it to have. And like I said, psilocin is very, very difficult to make just a prescription pill, tablet, etc. by itself, because it'll readily oxidize. And you can see here on the right-hand side of the slide, so we have the psilocybin that I've been talking about with this phosphate group. Let me point this out for us. But we have the phosphate group here that's removed by phosphatase enzymes, leaving us with psilocin, and you can see the very close resemblance between serotonin, our native neurotransmitter, and then the psilocin, which is really its reason for having the same level, or this high level of activity. So I'm going to go more into detail about this risk with 5-HT2B. This is something that is well established from other drugs, and all the classical psychedelics hit 5-HT2B. So psilocin does, LSD does, DMT does. Almost all of their synthetic analogs that have been made, a lot of these have analogs that are not relevant to what we're talking about today, but they all hit 5-HT2B. And so there's not a good solution that maintains activity at 5-HT2A, but then removes this 5-HT2B activity. And so from other medications, specifically some anti-obesity drugs from the 70s, 80s, and 90s, it's well known that 5-HT2B agonism over time will cause valvular fibrosis. And so this is something that's a fairly well-established cumulative effect. The more exposure you have there, the more likely you are to develop valvular fibrosis. And then once that starts, the more severe that it'll get over time with continued exposure. So the classical example of this is fenfluramine, or the commercial preparation, which is a mixture actually with another drug. Fen was very popular in the 90s, at least in the US and North America, and that led to significant litigation because we had a number of patients that developed valvular fibrosis, and this became a multi-billion-dollar, tens of billions of dollar litigation. So this is something that when you're designing a drug, you want to avoid absolutely as much as possible. And so the concern here is even to the level that the FDA last summer, for instance, put out a draft guideline for companies and groups that are developing prescription drugs in the psychedelic class, and their very number one bullet point on the things that they're concerned about is this valvular fibrosis issue. And so they want to see one of two things in regulatory facing clinical trials with these compounds. They either want to see that one, the drug doesn't interact with that receptor at all, therefore there's not that concern, or two, they want to see a very stringent additional layer of monitoring of patients, both with animal testing, looking for issues with the heart, as well as also monitoring of patients over time. And so this is a pretty big roadblock if you have that for a clinical trial program. So How have we actually done this? This is where I was going to, show you exactly kind of this is what we've done to address these issues. So we do have shelf-stable, heart-safe psilocin analogs, and you can see on the right, if you remember our psilocin, diagram from a few slides ago, what we've done is add in a nitrogen in the carbon ring. So we've done that at different positions. This is, like I was saying, we did this iteratively. So we knew that this side of the molecule was an area that we could work on. We knew that this side over here was the active pharmacophore, so we did not really want to touch that, but we could have multiple kind of tries on the same concept by putting a nitrogen in these different spots. And we know that adding a nitrogen into a carbon ring like this will usually create an additional layer of oxidative stability. So that was the first goal that we did that was successful. We've tested that in benchtop assays as far as stability. And then in testing this versus the serotonin receptors that we are interested in, we've seen also that this gives us selectivity against 5-HT2B at the same time. And so by being able to choose among these different 3 molecules, we can have both of those goals attained. We can have shelf stability, and then we can also have no activity at 5-HT2B. So the sort of take-homes with this is it allows for, like Rakesh I think mentioned nicely, the clinical trials are currently on just using this 1, 2, or 3 times. The durability does seem like it's for years at a time, but if you have somebody that has had lifelong depression, it's pretty practical to think that they may need to have a additional therapy every few years, every 5 years, every 6 months. It could be very, highly variable patient to patient. We don't really have a good idea of that, but this medically would allow you to have that repeated dosage and not worry about this cumulative risk. Also, there's incredibly strong interest, really globally. I mean, from Australia through Europe, through North America. I'm not sure about the interest level in Turkey at the moment, but I know sort of globally, there's this strong interest in microdosing, which is using these same compounds at a low dosage over an extended period of time. And a lot of times, because of the legalities of these compounds, this is done in a black market scenario where the patient is self-prescribing this to themselves. They go buy the compounds on the black market, and then they make dilutions or some sort of smaller preparation where they're taking a 1/10 or a 1/5 of what would be a normal recreational, psychedelic level dose on a daily or weekly basis. And so this is really concerning because these individuals out in the population, these citizens, have no idea about this heart disease risk. And it's a pretty big barrier for ethics boards looking at, can we actually do this in a clinical trial format? That's one of the reasons that we don't see microdosing moving into the clinical trials, is that we don't have great clinical data on whether it's actually efficacious. There's a lot of people that are evangelical about it. They say that it's fixed them, and they're very much proud about that, but it's not clear from the data. We have studies that suggest that yes, it's a good antidepressant, and then we have studies that suggest that it's placebo effect. So it's something that we haven't been able to fully address, but the population has really kind of already made their mind up and want this, and so this is something that, at least if that continues to play out on the side where it is relevant in the clinic, this would be a reasonable way to actually provide that as a long-term medication. And just from a conceptual level, it does kind of make sense that this would be a good alternate antidepressant. Your classical, most common antidepressants, the SSRIs, are making serotonin longer lived in the synapse. This is a very, very close serotonin mimic that you could boost just a little bit through dosage. So it does seem like they might be, in the long run, an interesting adjunct or alternate treatment, but that remains to be seen, absolutely.
Just Rob, just for Rob here, just for the audience and myself, where are you in terms of?
Oh, as far as the clinical trials?
No. In terms of towards ending your presentation?
Oh, I'm close to the end.
See, thank you.
So I'm going to go through this part relatively quickly, but these are the actual receptor binding assays that show this selectivity. Here you can see that in the 5-HT2B, we have 2 candidates compared to psilocin in the red that have no activity at 5-HT2B. So I'm going to end with the MDMA analogs that I mentioned earlier. So MDMA, its primary activity is through releasing neurotransmitters, specifically serotonin, norepinephrine, and dopamine. It also interacts with a lot of the serotonin receptors that we were just talking about from the classical psychedelics. It's kind of a mixture of both, but maybe 80% of the mechanism is through the release of neurotransmitters. Like we've covered, there's very strong evidence that this is very successful for treatment refractory PTSD, success in the 80s range. Also, this can be a therapy aid, but issues similarly to what we've been talking about are with duration, monitoring, the costs associated with that, and also the 5-HT2B risk that I just went over. So I think I can kind of go through this one fairly quickly because we've covered these parts, but you are looking at having 2 therapists monitoring a patient for 8 hours, so the cost there is really hard to control and scale. So a shorter acting MDMA would solve these issues. So this is how we've actually done that. Like I was saying, we can look at the structure of these drugs and then understand where they're actually active, and we can't change the structure, but then look at, okay, well, we can change oxygens for sulfurs, or we can change both oxygens for sulfur and nitrogen and actually get some of this. The logic behind this is actually kind of interesting. So we know that by adding a sulfur or a nitrogen, we can make this sort of side of a carbon ring more of a better substrate for known liver enzymes. So we're basically designing it so that the known liver enzymes can more quickly break down the compound and then clear it from the body. And so this would enable both a wide variety of use in therapy, PTSD, I think, being the best example, but also others, like I mentioned. It would allow repeated use because we don't have that 5-HT2B activity by also doing this again. And then the thing that I think is interesting is we've had discussions with just a number of people in the field about, well, what would you use these sorts of compounds for, considering that we've changed these properties. And one that keeps coming up is the use at a much lower than recreational dosage level, but as an acute anxiolytic for panic attacks, for phobias, this sort of thing. And so I think there's some interesting potential uses here. And this is the exact data that shows what we're discussing here. So we mimicked MDMA very nicely. This is the MDMA's effect on the serotonin receptors, so you can see it's active at all those receptors. You can then see here as well that we have no activity at 5-HT2B, which is this graph here with our compound of interest, one of the 2 that I was just showing you. And then this is, I think, the most important feature that we're seeing here is that if you look at culturing these 2 compounds, MDMA itself, or what we're calling 6-BZT, but one of the 2 analogs that I was just showing you, that we have a faster clearance over time in these liver cells. And so that's highly suggestive that systemically dosed in a human, you'd have about a half reduction plus or minus of the duration of the drug. So this is hitting that goal. And I think the peak amount, if you take a pharmacology or pharmacodynamics of MDMA and think of shortening it by 50%, that would give you this sort of goal of having the peak intoxication be a 1 or 2 hour therapy session, which is really exciting and addresses especially those scalability issues. So I'd just like to make a couple acknowledgments. So I'm the co-inventor on the molecules that are these new analogs that I was just showing you with Dr. Denton Hoyer. I'm the sort of biologist of the team, and he's the pharmaceutical chemist. The work could not have happened without the 2 of us collaborating on this, so I have a lot of thanks for working with Dr. Hoyer. I also would like to thank Applied Pharmaceutical Innovations, which is the group that operates between industry and academia that we've been doing all of this research with, and they're housed at the University of Alberta, and so a lot of the staff that's been working there and doing a lot of the synthesis, a lot of the data that I just showed you are within the pharmacy school at the University of Alberta. We've been working closely together for 4 years, and I couldn't be more thankful to have them as part of the team. It's been a really great effort there. And then finally, this is all work that's been done privately by Midas Innovations Group, so I just want to be clear about that. It's a commercial development with hopefully being able to get these into clinical trials in the near future. That's our current goal at the company, is to get that MDMA compound that I was just showing you into phase 1 clinical trials. So with that, that's the end of the presentation. I'm happy to take questions or discussion, so thank you for listening.